In 2020, the coronavirus pandemic reminded us of the fragility of the human condition. It demonstrated how little microscopic organisms may change vast microeconomic landscapes in a few months and profoundly shape our society. Throughout ages, humanity has been exposed to enemies which cannot be seen with the naked eye. However, for the first time in human history, we were better prepared than we have ever been. Since Watson and Crick discovered the double helix of DNA in 1963, molecular and cell biology has advanced tremendously. Nonetheless, the concept of quarantine was developed centuries earlier in response to a far deadlier and sinister disease. Its name? The plague. The plague was caused by the bacteria Yersinia pestis. The genus was named after the scientist who discovered it, Alexander Yersin, who joined the Pasteur Institute at just 22 years old. Biology is often written in our past. Such is the case with the decision of the Senate of the Republic of Venice to establish Lazaretto Vecchio as a quarantine island in 1423. The term quarantine was derived from the 40-day period during which sailors and merchants traveling to the Venetian ports were isolated. Not everyone survived, however. During the construction of a museum in 2007, archaeologists discovered mass burial graves containing over 1,500 plague victims quarantined there from the 16th to the 17th centuries. Why exactly 40 days? Well, to begin with, 40 is a number connected with purification in the Bible. Jesus was perhaps the first human to do a quarantena, a Latin term referring to the 40 days and 40 nights he fasted in the Judean desert. Moses, on the other hand, spent 40 days on Mount Sinai. A lazaret was not a novel concept. In 1377, in order to maintain trade between the Ottoman Empire and the West while preventing disease spread, the Republic of Ragusa in modern-day Croatia imposed a 30-day isolation period, Trentina, on people coming from affected areas. These places had one thing in common: flourishing trading routes. Venice became an important trading hub in Europe as maritime trade with the East expanded. However, exchanging goods and services implied the exchange of people, and the exchange of people implied the exchange of germs. Every day, boats from all over the world would converge in the Adriatic Sea. This was all that was required for the movement of one of humanity's most devastating pathogenic agents, Yersinia pestis. There are three major historic pandemic events, in fact: the Justinian Plague, the Black Death, and the Third Pandemic. Now, let's dive in and take Yersinia pestis apart with our molecular wrenches. Yersinia pestis is an aerobic bacteria that belongs to the Enterobacteriaceae family. This family is specially known for its members: E. coli, Salmonella, Klebsiella. Yersinia pestis optimally grows in temperatures between 28 and 30 degrees Celsius, but it can actually proliferate at a temperature range from 4 to 40 degrees Celsius. Notice how most pathogenic bacteria can grow at body temperature. Yersinia pestis evolved from a common ancestor shared with Yersinia pseudotuberculosis. During the Bronze Age, Yersinia pestis acquired the ability to be transmitted by ectoparasites such as the flea. Specifically, it acquired the Yersinia murine toxin, which allowed it to survive in the flea gut. One of the most terrifying ideas is that Yersinia pestis uses a biofilm to block the flea's foregut, preventing it from having a blood meal. As the flea starves, the number of bites increases dramatically, as does the likelihood of the bacteria infecting the host. This toxin, which prevents the digestion of Yersinia pestis inside the flea, is codified in a plasmid called PMT. Plasmids are small circular DNA molecules that can be shared between bacteria. DNA is a way of writing instructions to build proteins, molecular robots capable of performing specific functions. You can imagine it as instructions how to build a house or a ship using Lego. Some plasmids give superpowers to bacteria, such as resistance to antibiotics. Yersinia pestis harbors two other plasmids alongside with PMT that transform it in a true villain. The plasmid PCD1 is common to all Yersinia species that are able to cause infections in humans. It encodes important elements for infection, including a type 3 secretion system, which is a molecular syringe. Indeed, it transfers Yersinia outer proteins, or YOPs, to immune cells such as macrophages and neutrophils, disarming the immune system before it has time to respond. It also codes for V antigen, which further suppresses immune activity. The plasmid PCP1 encodes the protein plasminogen activator, a crucial virulence factor, as it promotes the spread of Yersinia pestis throughout the body from peripheral sites. In summary, after entering the host, Yersinia pestis spreads through the lymphatic system towards the lymph nodes. The growth of bacteria leads to inflammation, forming a bubo. Next, Yersinia pestis spreads from the lymph to the blood, thereby causing septicemic plague,